Polymerase chain reaction. Contents to be covered. Introduction. Basic components of PCR. Steps involved in PCR. Factors affecting PCR. Thermal cycle. Limitations of PCR. Applications of PCR. Introduction. PCR is a molecular biology technique used for amplification of DNA enzymatically and without using a living organism such as E. coli or yeast. Discovered by COAGULANTS in 1983. Based on using the ability of DNA polymerase to synthesize new strand of DNA complementary to the offered template strand. Basic components of PCR. DNA template, region of the DNA fragment to be amplified. 2 primers, these determine the beginning and end of the region to be amplified. Taq polymerase, copies the region to be amplified. Nucleotides, serve as the building blocks for the new strands. Buffer, provides a suitable chemical environment for the DNA polymerase. Major steps of PCR include denaturation, which means the separation of strands. Annealing, binding of the DNA primer to the DNA template. Elongation, addition of nucleotide sequences to the DNA primer. First step is initialization. This step is only required for DNA polymerases that require heat activation by hot start PCR. It consists of heating the reaction chamber to a temperature of 94 to 96 degrees Celsius, or 98 degrees Celsius if extremely thermostable polymerases are used, which is then held for 1 to 10 minutes. Second step is denaturation. This step involves heating the reaction mixture to 94 degrees Celsius for 20 to 30 seconds. During this, the double-stranded DNA is denatured to single strands due to breakage in weak hydrogen bonds. Third step is annealing. The reaction temperature is rapidly lowered to 54 to 60 degrees Celsius for 20 to 40 seconds. This allows the primers to bind to their complementary sequence in the template DNA. High concentration of primer ensures good yield. Elongation, also known as extension. This step usually occurs at 72 to 80 degrees Celsius, most commonly 72 degrees Celsius. In this step, the polymerase enzyme sequentially adds bases to the 3' end of each primer, extending the DNA sequence in the 5' to 3' direction. Under optimal conditions, DNA polymerase will add about 1000 base pairs per minute. Final elongation. This single step is optional, but is performed at a temperature of 70 to 74 degrees Celsius. The temperature range required for optimal activity of most polymerase is used in PCR for 5 to 15 minutes after the last PCR cycle to ensure that any remaining single-stranded DNA is fully elongated. Final hold. The final step cools the reaction chamber to 4 to 15 degrees Celsius for an indefinite time, and may be employed for short-term storage of the PCR products. Factors affecting PCR. Number 1. Primer. Primer are synthesized oligonucleotide usually ranging from 15 to 30 bases long. Primers are designed such that at 3' end they do not have more than 2 bases complementary to each other, as this results in primer-dimer formation. The G+C contents is in the range of 40 to 60%. The melting temperature of both forward and reverse primer is usually the same. Low concentration of primer results in poor yield, while high concentration may result in non-specific amplification. Hence optimal concentration of primer is needed, that is 0.1 to 1 micromolar. Number 2. Amount of template DNA. Optimal amount of template DNA usually in nanogram. Higher concentration inhibit or results in non-specific amplification. Number 3. Taq DNA polymerase. Taq DNA polymerase is 94 kilodalton thermostable DNA polymerase isolated from Thermus aquaticus. Optimal temperature for activity of Taq polymerase is 72 degrees Celsius, but it can tolerate high temperature and is not affected by denaturating temperature of 94 degrees Celsius. Stark DNA polymerase have both 5' 3' polymerase activity and 5' 3' exonuclease activity, but it lacks 3' 5' exonuclease activity, proofreading activity. Number 4. Reaction buffer. Recommended reaction buffer contains the following: tris HCl, which maintains pH. KCl, neutralizes the charge present on backbone of DNA. MgCl2, cofactor and catalyzer. Primers, determine the beginning and end of the region to be amplified. Nucleotide sequences, expand the growing DNA strand. Thermal cycler. Thermocyclers, or thermal cyclers, are instruments used to amplify DNA and RNA samples by the polymerase chain reaction. The thermocycler raises and lowers the temperature of the samples in a holding block in discrete, pre-programmed steps, allowing for denaturation and re-annealing of samples with various reagents. Limitations of PCR. Prior information of template necessary in order to generate primers. Like other enzymes, DNA polymerase is also prone to error, which may lead to mutations in the synthesized DNA. Even the smallest contaminating DNA would be amplified, leading to misleading and ambiguous results. After 20 cycles, PCR begins to slow down because of the limited reagent concentration. So, after a few cycles through 95 degrees Celsius, the DNA strands separated reattach to each other rather than annealing. Applications. Diagnosis of genetic diseases. Genetic counseling, screening the parents for genetic disease before deciding on having children. Pre-implantation diagnosis, screening for genetic disease before implantation of an embryo in IVF, in vitro fertilization. Screening for genetic disease before birth using tissue samples from the chorionic villus, the membranes found between the mother and unborn baby. Fetal tissue from the amniotic fluid, the fluid around the unborn baby, or the small quantities of fetal DNA, DNA from the unborn baby, found in the mother's bloodstream. Diagnosing inherited or spontaneous diseases, either as a result of symptoms, or because of family history, for example, Duchenne muscular dystrophy. Genetic fingerprints, paternity testing, tissue typing for organ transplantation. Detection and diagnosis of infectious diseases. Detection of infection in the environment. Personalized medicine. PCR in research, analysis of gene expression, for example looking at levels of expression and when genes are switched on and off in physiological processes, including in health and disease. Other uses, archaeology, to identify human or animal remains. Degraded DNA samples may be able to be reconstructed during the early cycles of PCR. Differentiate between similar species of organism. Thank you for watching.